Thank you for inviting me here. The title of my lecture is Molecular Noise, Non-stationarity and Memory in Single-enzyme Kinetics. So I'll just give you the background of the work I'm going to present. So in 2006, there was an experiment done in Harvard in which the effect of molecular noise in single-enzyme kinetics, or more specifically in Michaelis-Menten kinetics, was measured. So the title of the paper was Ever Fluctuating Single-Enzyme Molecules Michaelis-Menten Equation Revisited. So in this experiment, several non-classical effects were observed which could not be explained or which cannot be explained using theories based on classical deterministic kinetics. So over several years, we have developed a new time-based approach, a new time-based stochastic approach, which in combination with a stochastic number-based approach, which is namely the chemical mastery equation, unifies the results of single molecule stochastic and classical deterministic enzyme kinetics within a single theoretical framework. So in my lecture today, I'll provide a new method of statistical analysis to understand enzyme kinetics at the molecular level. So I'll first like to acknowledge this work was done with my former PhD students Shoma and Ashutosh at IIT Madras, with Shomdev at IISc Chennai, and Professor Ranjay Adhikari at DAMT, University of Cambridge. So here is the outline of my presentation. So I'll first give a brief overview of deterministic and stochastic Michaelis-Menten kinetics. I'll then present or summarize the results of single enzyme experiment. I'll then present our generalized theoretical framework based on the count and point process description of stochastic processes. In comparison with experimental data, new theoretical predictions, summary, and conclusion. So enzymes are biological catalysts or molecular machines that carry out the conversion of substrates to products without getting consumed in the process. So the question of relevance in enzyme kinetics has been, what is the rate at which this process is carried out? Or what is the enzymatic velocity for this process? So the answer for this was first provided by Michaelis and Menten in 1913 for a deterministic network of 3 elementary reaction steps. So in this reaction, enzymes react with substrate to form complex, which can dissociate to form product while regenerating the same enzyme. The steady state velocity for this reaction is defined in terms of the long time limit of the rate of product formation. Now this quantity can be obtained from deterministic rate equation. By solving deterministic rate equation, we can calculate what this dp dt is. It turns out that this is given by this expression, and this expression provides a fundamental hyperbolic relation between substrate concentration and steady state velocity. And this is called the classical Michaelis-Menten equation, and this happens to be one of the most celebrated expressions in biochemistry. So these deterministic rate equations are obtained from, are written in terms of rate of change of enzymes, complexes, and products, and they implicitly assume that enzyme concentration is thermodynamically large. That means when we are writing this equation, we implicitly assume that n is approaching infinity, where n is the number of enzymes. So it is in this limit the Michaelis-Menten expression is derived. So this is the Michaelis-Menten mechanism is a cyclic mechanism in which the rate of product formation rate determines the enzymatic turnover cycle. So this rate equations for these mechanisms are linear ordinary differential equations. This is because of the pseudo first order approximation in which the bimolecular second order complexation step is replaced by the pseudo first order step, with an effective rate constant Ka, which depends on substrate concentration. So the rate equations for this mechanism can be easily solved, and they give a solution, a stationary solution, when time is beyond this short transient limit, which is defined by the rate constants of the reaction. Here Ka is K1S and Kb is the inverse rate constant, is a rate constant for dissociation of complex given by K minus 1 and K2. So it's a sum of K minus 1 plus K2. That is Kb, this is Ka. The short transient regime that is observed in classical enzyme kinetics is defined in this term. So for time beyond this short transient, which is Ka plus Kb inverse, these two enzymes and complexes are in equilibrium, and this equilibrium condition, a classical equilibrium condition, is the Michaelis-Menten expression. So this is how we can derive it using the rate equation for formalism. The Michaelis-Menten actually derived it differently. They used what is known as a fast equilibrium condition, in which a timescale separation is assumed between the fast equilibration of enzymes and complexes and slow product formation from the equilibrium ensemble of complexes. So in this mechanism, or in this condition, assumption, the network is broken down into an equilibrium mechanism in which E and Es are in equilibrium with each other, and a slow product formation step in which the rate of product formation depends on this equilibrium ensemble of complexes. So this is the fast mode of the kinetics, this is the slow mode of the kinetics. So this automatically gives this equilibrium condition, and from this equilibrium condition, Michaelis-Menten equation can be derived. From this, one can obtain equilibrium enzyme substrate concentration, which is given by this, and together they give the steady state velocity at the onset of the reaction. That is when the time starts, you can get the steady state velocity and the enzymatic velocity. So this is the hyperbolic Michaelis-Menten expression, and here in this mechanism, equilibrium is realized at the onset of the reaction. And this is what is also observed experimentally using the initial rate method used by Michaelis and Menten. So the hyperbolic dependence of substrate or velocity on substrate yields a linear relation between the inverse of velocity and substrate concentration. This is what is shown here. This is the hyperbolic relation between velocity and substrate concentration. And if the inverse of V and 1 by S is plotted, you get a linear relation. And kinetic data for large majority of enzymatic reactions, when fitted to this linear form, is rate parameter of the Michaelis-Menten mechanism. So there are two important rate parameters for the Michaelis-Menten mechanism. One is the Michaelis constant, which is the ratio of the dissociation divided by association rate constants, and Vmax, which is the maximum velocity that an enzyme can have. And this limit is realized at high substrate concentration. V equals to Vmax. At low substrate concentration, it is linear. And there is a crossover from linear relation to this constant velocity, which is Vmax, with the increase in substrate concentration. So it turns out that if you re-express this velocity, the hyperbolic dependence of enzyme velocity on substrate, in this more generalized form, or more generalized input-output form, this expression appears in many different places in chemistry and biology, including adsorption, surface adsorption, bulk enzyme catalysis, single molecule catalysis, transcription, translation, kinetics of linear motors, rotary motors, chromatin remodeling, so on and so forth. So this table has been taken from this reference, where the ubiquity of the Michaelis-Menten expression in diverse chemical and biological contexts has been rationalized by a graph theoretic method. And this method uses the graph-based linear framework for time-scale separation between the slow and fast modes for deterministic enzymatic networks of complex topologies. So this is not only true for Michaelis-Menten mechanism, which is a simple mechanism, but even for a mechanism which is more complex, we see that the hyperbolic relation is a general result of Michaelis-Menten. And if we can divide the network into slow and fast modes, we end up getting a hyperbolic relation. So it's a very general result. So under physiological conditions, however, enzyme concentrations are not thermodynamically large. Typically tens to thousands of enzymes catalyze thousands to millions of substrates. So the question is the Michaelis-Menten expression a valid expression or valid description at such low enzyme concentration or at the single enzyme level? So to understand this, or even to answer this, we need to understand a few facts about enzyme kinetics at the molecular level. First, that enzymatic reactions at the molecular level do not proceed deterministically. The fluctuations of both quantum mechanical and thermal origin, together termed as molecular noise, which are inherent to reaction catalyzed by individual enzymes. Because of the presence of this molecular noise, each step of the mechanism is stochastic, implying that neither the lifetime of a given state and nor the state to which it transits can be known with certainty. The effect of fluctuations and thus the uncertainty diminishes progressively with the increasing number of enzymes and vanish for their bulk amount. So in the thermodynamic limit of n going to infinity, the classical deterministic rate equations are recovered. So what is the main difference between enzyme kinetics at the molecular level and classical enzyme kinetics? So in enzyme kinetics, products are formed together in bulk, and one can define their concentrations, one can define their rate of change. In stochastic enzyme kinetics or single enzyme kinetics, products are formed one by one in discrete turnover cycles. One cannot define concentrations and one cannot define their rate of change. So this is a typical single enzyme Michaelis-Menten network in which a single enzyme reacts with a substrate to form a complex. So this complex and enzyme can fluctuate between one another before forming a product and regenerating the same enzyme. So the regenerated enzyme starts another turnover cycle, say the second turnover cycle, and again reacts with the substrate and form a complex, and then form the second product. So this process goes on. So at the end of the pth turnover cycle, there will be p products. So enzyme kinetics at the molecular level is described in terms of three time scales. One is the lifetime of E and ES states, the waiting time between two consecutive product formation, labeled here as tau 1, tau 2, tau 3, tau 2, and so on, where p is the turnover number, which takes the integer values starting from 1, 2, 3, and so on. So tau 1, tau 2, tau 3, tau p are the waiting time between two consecutive product formation, and capital TP is a turnover time for the pth product formation starting from time t equals to 0. So this is the first turnover time, this is the second turnover time, and this is the pth turnover time for the reaction. Now, these times are not independent, they're related to each other. The waiting time of the pth product is the sum of the lifetime of E and ES. As they're fluctuating, the fluctuations are accounted for, and capital T is the turnover time for the pth product formation, which is the sum of the waiting times, and therefore is a sum also of lifetime of E and ES states. So all these times are stochastic quantities with well-defined distributions. So enzyme kinetics at the molecular level is described in terms of the distribution of waiting times and their waiting times or turnover times and their moments. So there's a hierarchy of distribution depending on whether this waiting time depends on 1 time, 2 times, or 3 times, and so on. So the mean of the first of 1 point distribution is the mean waiting time. Its variance is waiting time dispersion. Its the first moment of this is waiting time correlation, and so on and so forth. So in this work that I'm going to present, we provide a general theoretical formalism to calculate all these kinetic measures of interest. So the quantities which are listed inside the table have been measured in a recent experiment, which I'll talk about now. So this experiment, as I said, was done in 2006, and it had single-enzyme kinetic, so single-enzyme kinetic measurement, which has single enzyme and numerous substrates. So in this experiment, the turnover kinetics of a single enzyme of beta-galactosidase was measured using fluorescence spectroscopy. Here, enzyme and substrates were non-fluorescent, and product was fluorescent. So waiting times between two consecutive product bursts was recorded repeatedly under identical conditions, and its waiting time was obtained. And this yielded a distribution of waiting time called the waiting time distribution. So here is the distribution which is obtained experimentally, and it shows multi-exponential decay. It shows multi-exponential decay here in red lines, sorry, in blue lines. So, so this is the plot from waiting time distribution. The two things to note about this experiment: first, that beta-galactosidase is an enzyme with 4 independent and identical binding sites and is known to follow the Michaelis-Menten kinetics in bulk. The second is that in this experiment, the enzyme was immobilized on a glass surface, and therefore the effect of diffusion was suppressed. So,
what are the discrete points in your previous slide? What are the discrete points? What do they represent?
These one.
Yeah, all the discrete. You have continuous lines and discrete points.
So these are not my points. So these are the graph taken from the experiment, and what they have done is that they have measured the waiting time as a function of time. So this is, I think, experimental error perhaps, which is being accounted for. Then they have done some theoretical analysis, and this line represents that theoretical analysis, about which I'll talk about. Okay.
Very fitting to the data.
Yeah, it doesn't seem like a good fit. That's why I was.
So what was the role of that? There are 4 of these binding sites.
Yes.
Does it play a special role?
It plays a special role in our theory, not in the theory which was done after this experiment was. There was a theory which, so this fitting that they have done had no role. So yes.
That waiting time distribution.
Yes.
Or the turnover time distribution, rather.
Yes.
The turnover time distribution that was measuring irrespective of whether this particular one,
Yes.
for example, came from the right or left or the upper or the lower. Right?
Yes. Yes.
And so simultaneously, when a product is coming out,
Yes.
it's possible that another substrate is bound to another binding site simultaneously.
The substrate is bound to another.
Can simultaneously
Yes.
4 substrate molecules can bind with it at different stages of their enzymatic cycle?
They can, but in most theories, based on single enzyme kinetics, that aspect is not taken care of.
But in the experiment, it is possible.
It's possible that there are 4 sites, and you're saying that all those 4 sites are occupied, are bound with substrates. It's possible, but you have to take it into account in theory, which hasn't been taken into account. So I'll just tell you the kind of theories people have been doing with time. I'll just tell you as I proceed that single enzyme theory for them is a single enzyme, single molecule. So that is how they do theory, but we have taken it into account in a different way. But I'll just talk about that. Yeah, I'll come. So I'll explain.
Can I ask one question?
Yes. Yeah.
So you have different reaction rates. Okay, so you know the average time required for the reaction, that is inverse of the reaction times, right? So can one write down the expressions for the waiting time distribution in terms of those reaction rates?
We don't know the waiting. You see, we know the mean waiting time. Is that what you say?
Yeah.
We don't know. So we're going to calculate. First of all, we'll get the distribution from a theory, and then we'll calculate it. And I'll just tell you what has been done and what we have done differently. So, so this is what I just, in the moment, still talking about the experiment. So what they observed was that the inverse mean waiting time is gives the Michaelis-Menten or Michaelis-Menten-like expression So is Michaelis-Menten-like expression is that you'll still get a hyperbolic relation, but Km will be different and K2 can be different. So there will be effective rate constant which will be different, but the expression will be hyperbolic. So this is what they get. So you inverse of the mean waiting time will give you Michaelis-Menten or Michaelis-Menten-like. I'll again come back to this, and please remind me if I don't tell you later that what is Michaelis-Menten-like. So the Michaelis-Menten, so this is the relation which inverse of the mean waiting time observed from the distribution obtained experimentally gave the Michaelis-Menten equation for a single enzyme. And so this relation is based on the observation that the Lineweaver-Burke plots for both single enzyme and bulk amounts were linear and showed data collapse when plotted as a function of mean waiting time and inverse substrate concentration, and inverse velocity and inverse substrate concentration. So they both showed linear curve and they showed data collapse for different, and therefore they can be related. But the substrate concentration range was very low for which the measurement was done. So the conclusion, the inference is that the Michaelis-Menten or Michaelis-Menten-like equation is obeyed at very low substrate concentration. So this is the measure of mean. The measure of fluctuations included randomness parameter, which is the dimensionless variance of the same distribution I showed. Then 2-point waiting time distribution, which depends on 2 times, and 2-point waiting time correlation given by this. Now, this was found to be greater than 1. Randomness parameter, which I said is the dimensionless variance of the distribution, was found to be greater than 1. And this is a signature of dynamic disorder. Again, I'll explain what it means. And 2-point waiting time distribution yielded molecular memory, in which long first waiting time is likely to be followed by long second waiting time, or short first waiting time likely to be followed by short. So this is long, short kind of memory, which is positive. And the other thing that they found was the waiting time correlation, which was found to be positive, implying that turnover times are statistically dependent. So the question is, how can molecular memory and statistical dependence between consecutive turnover events emerge from the fundamental memoryless character of elementary reaction steps, which is what is dictated by the mass action kinetics. So, so to answer this, we provide a generalized theoretical framework that unifies the results of deterministic and stochastic enzyme kinetics. And this framework is based on the chemical mass equation approach of stochastic processes or stochastic kinetics. So the CME, in short, accounts for the molecular discreteness and stochasticity in each elementary step of the reaction mechanism. It is written for the time evolution of probability distribution of number of enzyme complexes product at a given time. The right-hand side shows the transition probability of going from E to ES state. This is the first term here. This transition probability of E to ES state, in which number of enzyme is going to decrease by 1 and complex is going to increase by 1 as a result of the forward reaction step. So this is transition probability of the backward step, and this is the probability of the irreversible step, in which number of enzyme substrate is going to decrease by 1 and product and enzyme is going to increase by 1. And this is written with respect to this initial state here. So this is the CME for the Michaelis-Menten mechanism. And in the limit of n going to infinity, which is the thermodynamic limit, it recovers the deterministic rate equation. This was shown by Bartholomew in 1962, and this is because, the CMA describes a discrete state continuous time microprocess in which the lifetime of each state is exponentially distributed, and that's why we get back the deterministic rate equations. So stochastic trajectories obtained from chemical mass equation specify the discrete change in species number as a function of time. So the plot is shown here, and this can be analyzed in 2 ways called the count process or point process descriptions of stochastic processes. So count process description counts the number of products formed in a given time, and point process description specifies the time required for the pth product to form. So in most earlier studies, count process has been used to analyze enzyme turnover statistics. So this is because chemical mass equation naturally gives count process description in terms of the probability of number of species in the reaction. The solution of the CME naturally gives the probability or the joint probability of the number of species in the reaction from which the probability of number of products at a given time can be obtained. So once we know this probability, we can obtain mean velocity, and one can obtain what is called the Fano factor, which is the number correlation or the dimensionless number correlation. So one can get from mean and variance of the distribution, mean velocity and number fluctuations. So this description is relevant in the classical limit of large number of enzymes and products because it is in terms of numbers or mean numbers. So what we do is, or what is more important is that for in the point process description, which specifies the time required for the pth product formation. So point process will give the turnover time distribution or waiting time distribution, the mean of which gives the mean time and the variance of which gives the variance of the distribution or the dimensionless variance of the distribution called the randomness parameter. So this description is relevant to single enzyme kinetics, and the question is, how do we go from this count process description to point process description? So, so a few years back, we solved the chemical mastery equation for the Michaelis-Menten mechanism exactly for the first time. So this is the solution, and we use the generating function method. And using this, we found a relation, a sought after relation between the count and point process description. And we again use this to get exact expression for the turnover time distribution for any n and any p, where n is the number of enzymes and p is the turnover number. So this is the point process description, and this is the waiting time distribution or turnover time distribution, which is expressed in terms of the count process description. This is the solution of the chemical mastery equation. So from this, we can analytically calculate the waiting time distribution for any n and p, as I said. And this is the generating function solution, which we can substitute here to get turnover time distribution. So this is the analytical expression that we can get, which are exact. Higher order waiting time distributions and correlations were obtained from the Gillespie simulation, which generates exact numerical trajectories of the chemical mastery equation. So now I'll show results. So for n equals to 1, we find that this is the waiting time distribution that we obtained from CME. Or more specifically, it's the turnover time distribution because it depends on capital TP. So it's a turnover time distribution from which we get the first turnover time distribution for n equals to 1, which, as I said, is given by this, which has a peak distribution. It decays exponentially and rises up exponentially. So this is the distribution for n equals to 1, p equals to 1. We find the inverse mean of the distribution, and it gives us the Michaelis-Menten equation exactly for n equals to 1. So more generically, if we compare this waiting time distribution for higher p, we find that waiting time distributions are independent and identical for n equals to 1. That means they don't depend on the turnover number. So this is the waiting time distribution. That means the turnover statistics is only determined by one time scale, which is given by tau. So the inverse of this mean waiting time exactly recovers the Michaelis-Menten equation for n equals to 1. And this is the point process description of single enzyme velocity because it is in terms of mean time. So this is a measure of mean statistical measures of fluctuations, including randomness parameter, which is the dimensionless variance, and it was found to be always less than equal to 1 for all s. waiting time correlation between first and subsequent turnovers were found to be 0 for this case, for n equals to 1. So what this suggests is, all these results for n equals to 1 suggest that waiting time distributions are independent and identical, implying that single enzyme turnovers follow the renewal stochastic process. What it means is that the mean pth turnover time is equals to p times mean waiting time. So this is related perhaps to your question, that we know that the mean waiting time is for the process. We are actually getting it from the distribution, and for single enzyme, it turns out that these are equal. So all these, between two consecutive products, the mean time is the same, and this is a result of renewal theorem.
But this is because they are independent and identically distributed.
Yes, so this is coming from renewal theorem, yes. So the inverse of this mean waiting time, as I said, gives the Michaelis-Menten equation, but using this equation, it can be expressed as P by Tp from here, and this gives correlation 0 as they are independent and identical. So this is a result for single enzyme, and I'll come back to this if I have time. So this is this is a randomness parameter. So this is the squared. Yes. Yeah. Yes. They're not. So I'm going to tell this. The story, I'll just, yeah. So I just come back to this slide if required later. So this is a randomness parameter, and this is the dimensionless variance of the distribution. It's also called the squared coefficient of variation. So for an exponentially distributed reaction, like for a single step reaction, when the distribution is exponential, the variance is equals to mean square. And for this reaction, R is 1. So this randomness parameter is generically 1 for waiting time distribution that decays exponentially. If it decays multi-exponentially, then R is greater than 1. And if it has a peak, R is less than 1. And if this peak has larger variance, then R is much less than 1. So this is a general result for single enzyme kinetics. So what is another thing which is known is that the substrate variation of the randomness parameter links the magnitude of intrinsic temporal fluctuations with network topology. So if the network topology is linear, then the distribution is peaked, and therefore R is less than 1. If it's a single step reaction, then it decays exponentially, and then R is 1. But if a network topology is branched and it has multiple competing reaction pathways, then R will be greater than 1. So this is a general result again, which is very important for single enzyme community because this is a measure of intrinsic temporal fluctuations which are large. So dynamic disorder is an emergent property of a single enzyme network with branch topology based on any theory which is based on Markovian theory will give R greater than 1 or less than 1 depending on if the network is branched or it's linear. So for a branch network, when there are multiple competing pathways, R is greater than 1. If the rate parameters are such that the linear pathway is followed within a branch network, then R is less than 1. For linear network, this is always less than 1. So no dynamic disorder for linear network, and this is greater than 1, this is less than 1. I'll just combine the results now. And so for single enzyme kinetics, what we find is that inverse of the mean waiting time recovers the Michaelis-Menten equation. This is the classical enzyme velocity, this is single enzyme velocity, and together they give the classical Michaelis-Menten equation when you combine the point and count process description. So statistical measures of fluctuations was found to be less than, because Michaelis-Menten mechanism is linear network is less than 1, therefore it doesn't show dynamic disorder. And correlations were found to be 0 and therefore no molecular memory. So all these results, at least both these results, suggest that single enzyme kinetics cannot recover experimental results. So next we considered n greater than 1, and we have this distribution here, which is the distribution now, which is multi-exponential, and we find the inverse mean of this distribution, or inverse mean turnover time for this distribution. and it does not follow the Michaelis-Menten equation. For p less than p star, I'll just come to that point. So for n equals to 2, for example, we can exactly get what the equation will be. This equation is not the Michaelis-Menten equation. This is for n equals to 2, the velocity dependence on substrate, and it's not hyperbolic. So again, generalizing this, we find that for n equals to 1, waiting time distributions depend on the turnover number for lower turnover numbers, but become independent of the turnover number for higher turnover numbers. So which means the turnover statistics is non-renewal for p less than p star, and it is renewal for p greater than p star. Here p is the critical turnover number. So this is the enzymatic velocity which we define, and this is the ratio of turnover number ratio of turnover number and mean pH turnover time for n enzymes. And we plotted substrate variation, and we find that for p much less than p star, this velocity depends on the turnover number and does not obey Michaelis-Menten equation. For p much greater than p star, the Michaelis-Menten equation is exactly recovered. We called it the transient regime, and this is the steady state regime. The question is, what is p star? So to answer that, we obtain two more functions. One is the waiting time correlation, 2-point waiting time correlations, and 2-point waiting time distribution. So 2-point waiting time correlation was found to be negative for p less than p star, but was equal to 0 for p greater than p star. The 2-point distribution yielded molecular memory, in which long first waiting time is likely to be followed by short second waiting time. So the memory was long short type. It was negative. So we got negative correlations and negative memory. So we infer that p star is a critical turnover number above which the waiting time correlations are absent and memory is lost. So for p less than p star, we are in the non-classical transient regime where turnover times are correlated. The time scale separation which is assumed in deterministic kinetics is not valid, and the Michaelis-Menten equation is not obeyed. For p much greater than p star, we are in the steady state regime where turnover times become uncorrelated, time scale separation is valid, and Michaelis-Menten equation is recovered. So to summarize, the stochastic kinetics yield negative memory and negative waiting time correlations for multiple enzymes, and no memory, no correlations, and no dynamic disorder for a single enzyme. So both these results don't explain experimental data, and I'll again come back to this slide. So now we find that single enzyme Michaelis-Menten kinetics cannot recover experimental results. So we need to include two important aspects of enzyme kinetics at the molecular level. First, that an enzyme can change its conformation during the course of a product turnover cycle. That these are like conformational fluctuations, and that most enzymes have multiple binding sites. Beta-galactosidase, for example, has 4 independent and identical binding sites, which has not been taken care of. So we include these two effects in the parallel pathway Michaelis-Menten network, which we in short call as PPMM, and which in its minimal form has an enzyme which can fluctuate between two conformational states before forming a product through either one of the two possible pathways. So there's one enzyme which fluctuates between two states, and it can form product through either one of the two possible pathways. So in the absence of conformational fluctuations, we get back the Michaelis-Menten equation. Michaelis-Menten mechanism. So this is a model for single site with single enzyme. For multi-site model, like single enzyme with multiple sites, we consider n replicas of this model. So we call it multi-site model or replica network. And we then solve the chemical mass equation for both these models and compare results with experimental data. So we find that the single site model can reproduce mean and variance of the distribution but cannot yield molecular memory because there are no correlations. Multi-site models can recover both the mean and variance of the distribution and can yield memory and correlations, which are positive. So in short, a multi-site model yields excellent quantitative agreement with single-enzyme data for the microelement kinetics. It quantitatively reproduces the mean and dimensionless variance of the waiting time distribution. It gives waiting time correlations which show stretched exponential decay. This is in quantitative agreement with experiment. This is a stretched exponential decay and recovers correct sign of the molecular memory, which is long, long, short, short. So this is the table which summarizes the result, and it shows that only PPMM model with 4 binding sites can recover all the experimental results, including positive memory, positive correlations, and randomness parameter with dynamic disorder. Microelement model with single site cannot recover experimental results. With fluctuations or without fluctuation, it does not. So the question is, what is the source of molecular memory in multi-site models? So to answer this, we carried out a detailed theoretical study, and we found that in the presence of multiple sites, there emerges a new time scale called the recurrence time. And this recurrence time makes the pth waiting time conditionally dependent on the p minus 1th turnover time. Okay, so I'll just explain this using this picture here. So I'll consider replica networks of 2 sites shown in red and blue color. Each of these sites can form products independent of one another because these sites are independent. So it forms products independent of one another. But what experimentalists are observing is the pooled output of these products, irrespective of where these products are formed. I think this was related to your question. So it doesn't matter where the products are formed, experimentalists are only observing the order in which these products are appearing, independent of where they originate from. So this is the pooled output which is being observed and which has been shown as superscript 2 because we have pooled 2 processes, 2 stochastic, independent stochastic processes into one output which is given by this pooled output, green color. The first waiting time of this pooled output is the waiting time of the first waiting time of a blue colored site. The second waiting time is the recurrence time, which is shown in this green color, tau plus. This recurrence time depends conditionally on the p minus 1th turnover time. Okay. So this is what makes this turnover time for the pth waiting time conditionally dependent on the p minus 1th turnover time. So we have this equation which takes into account what I just explained for the pooled output of new sites. Nu is greater than 1. If nu is equals to 1, then there is no conditional dependence and no memory. For nu greater than 1, there is conditional dependence and memory. So we take the time derivative of this and we find the waiting time distribution for the pooled output, which is given by this expression, and this depends on the waiting time distribution of recurrence time. So this positive is for the recurrence time, and this is called actually distribution of forward recurrence time, which was first given by Cox in 1962 in his book. So this distribution depends on velocity memory kernel, and this is the waiting time distribution for a single enzyme, and this velocity, when it approaches its steady state value, then the memory is lost, which shows that this memory is a transient effect that is being observed experimentally. So this is the velocity, and this is the plot of, it is a heat map depicting conditional dependence of pth waiting time on p minus 1th turnover time obtained from this equation, and it shows that there is memory for P less than P star, and there is no memory for P greater than P star. So again, it's showing that the emergence of molecular memory is a transient effect. So after this, we introduced new statistical measures to distinguish between the stationary and non-stationary states in single-enzyme kinetics. And this we do by combining the statistical measures of count and point processes description for replica networks. By replica network, we mean a pooled output of products generated from new greater than one sites, IITs, independent and identical sites. So this is the count process description, and this distribution, this yields steady state velocity for new sites and Fano factor for new sites. This is steady state Fano factor, steady state enzymatic velocity. So these are the statistical measures of stationarity that we obtain from the count process description. For the point process description, we introduced two new measures. One is pH dependent enzyme velocity, another is pH dependent randomness parameter. And these two new statistical measures can be applied to both stationary and non-stationary states in single enzyme kinetics. So how do we relate this with this? So how are they linked? So this is the pH dependent enzymatic velocity, which is the ratio of P and mean pH turnover time for the pooled output. For P greater than P star, this becomes independent of P because of the renewal statistics, and it's given by P divided by nu tau. And these results show that this Vp becomes independent of P and becomes equals to this quantity which doesn't depend on P. So this is the steady state velocity obtained from the point process description. This is the steady state velocity from the count process description, and the equivalence between the two processes yields the steady state velocity and gives the condition of stationarity for mean velocity at the single enzyme level. So this is randomness parameter. This is pH dependent randomness parameter, and this is given by the dimensionless variance of the turnover time distribution. And this depends on correlations. So when we are in the limit of P going to P greater than P star, these correlations are absent and randomness parameter become independent of the turnover number. And it becomes just the dimensionless variance of the waiting time distribution. So this is the randomness parameter, which is stationary. And this is again the point process description of temporal fluctuations, which we relate with a steady state Fano factor. So this is the equivalence between the point and count process descriptions of number and temporal fluctuations in steady state. This is the condition of stationarity for temporal and number fluctuations in the steady state for single enzyme kinetics based on pooled output. So just like to mention that a special case of this relation, which we have derived from the renewal theorem, was considered in this paper. So a special case of this for nu equals to 1 was first considered by Bloch and Schnitzer in the context of molecular motors, where it has widespread application. So this is the randomness parameter for one site, and this is the steady state Fano factor for one site. For P less than P star, these equalities are not followed. So this is what defines the non-stationary state. So next, we use these conditions of stationarity to estimate the critical turnover number from single enzyme data. And we do this by, first of all, getting these fitting parameter that we had obtained by comparing our theory with experimental data. So these are the model parameters estimated from a simultaneous least square fit of mean turnover time and randomness parameter to experimental data. So we use these parameters to obtain pH-dependent velocity for 4 sites, and this is the expression, and we study its variation as a function of substrate concentration. So experimental data is available for very short range of substrate concentration, which lies here, which was inconclusive in, predicting whether this is non-hyperbolic or hyperbolic. So what we do is that we take these fitting parameters and extrapolate to higher substrate concentration. And we find that when we, at high substrate concentration, this fitting give us non-hyperbolic velocity for what was fitted by them. So this is non-hyperbolic, and hyperbolicity and stationarity is recovered for P star around 50. So above P star, the stationarity is attained in terms of the equivalence between the point and count process description of velocity. So this is one result. And then we again use the same parameters. So we again use the model parameters which we had obtained from simultaneous least square fit of theory with experimental data to determine peer-dependent randomness parameter and peer-dependent Fano factor. So we plot this randomness parameter as a function of P and Fano factor as a function of time. And stationarity is attained again for P star around 50 for this model. So the stationarity is in terms of the equivalence between the point and count process descriptions of intrinsic, temporal, and number fluctuations. So again, combining these results, we get that these are the conditions of non-stationarity in which memory is there and therefore the enzymatic velocity is non-hyperbolic. These are the conditions for stationarity in which memory is lost and hyperbolicity is recovered in terms of the enzymatic velocity. So the fading of memory, the convergence of the waiting time distributions, and the equality of statistical measures all occur at roughly P star around 50 in the PPM model. So this is a summary of our results. So what we have done is we have generalized the Michaelis-Menten formalism to all n time. N is the number of enzymes. What we have known is that in the classical limit, Michaelis-Menten equation is realized at the onset of the reaction, and the transient regime is too short to be observed. So the Michaelis-Menten equation is recovered in the steady state, which is realized at the onset of the reaction. This is what we have known. What we find is that for n equals to 1, there is no non-classical transient regime, and Michaelis-Menten equation is obeyed because it obeys the renewal theorem. For n greater than 1, there emerges a non-classical transient regime in which turnover times are correlated. There is molecular memory, and Michaelis-Menten equation is not obeyed. And for P much greater than P star, steady state is realized where turnover times are uncorrelated. Michaelis-Menten equation is obeyed, and this is true for finite n in long time limit. So based on these results, we infer that single enzyme experiment on Michaelis-Menten kinetics of on beta galactosidase capture the results of the transient regime and not the steady state regime. So these are the concluding remarks. So these works show through a new method of analysis that memory and non-hyperbolicity have a common source in an observably long transient peculiar to stochastic reaction networks of multiple enzymes. The transient yields asymptotically to a steady state in which memory vanishes and hyperbolicity is recovered. So we propose new statistical measures to distinguish between the non-stationary and stationary states in single enzyme kinetics, apply them to experimental data from the landmark experiment that first observed molecular memory in single enzyme kinetics. And the mechanism of molecular memory presented here can be viewed as a null model in the sense of null hypothesis that should be tested against before embarking on a research for more elaborate models of molecular memory. So I have 5 minutes and I'll take your questions. I have a few more slides, but I'll take questions first. Yes. 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 Yes.
So then if one measured
Yes.
the distribution of the turnover times,
Yes.
then these effects that you are showing, memory and so on, so these will disappear?
I think so, yes. Because then they can distinguish, and then it should be like single enzyme experiment. This is what I think. What is thought to be a single enzyme experiment is that you have single enzyme. Here we have taken 4 sites which are independent and we are getting memory out of it. But if they can be distinguished in terms of 4 colors, I think you'll get back their no memory picture, I think.
And these four sites, when the substrate are bound,
Yes.
how one makes sure that they are actually not influencing each other? The reason I'm asking
Yes.
is that when you have this cooperative
Yeah, exactly.
enzymes, you
Yes.
get Hill equation
Yes.
and not Michaelis-Menten, right?
Okay, so the only answer I can give you is that when there's Michaelis-Menten equation which is being followed, there should not be any correlation. So when cooperativity is there can be correlation in the steady state, because just because you're saying that the velocity itself will be different and there can be correlations, but those correlations will have different origin, not the ones we are theorizing.
I was surprised that in spite of the fact that there are these four binding sites and they are all doing this enzymatic activity.
They're actually independent in this case.
Independent.
Yes.
How they remain independent, I find somewhat surprising, right?
I mean, how are they? How? Okay. So you're saying how independent are they?
No, actually, in spite of the fact that there are four binding sites,
Yes.
so they cannot be too far from each other. And so when there are conformational changes, it's surprising that one conformational change at one location
Yes.
is not affecting other three.
Yes. So yes, it's actually surprising. So the thing is that the only as a theorist, I think the only thing we can say that uh, if there is Michaelis-Menten and there are correlations, then I think we can at least infer just from the very beginning that they cannot occur simultaneously. So correlations had to belong to transient. So this is what we are saying. But what you're saying is that if there's cooperativity of some sort, even if it's weak cooperativity, you will see something different from this. And I agree with that. Yes, yes, yeah. So I don't know. I can't answer that. What they have observed, because I don't know how this thing, how much they interact with each other is not known. No, these correlations are non-stationary because they're not there in the steady state. So that we have checked. So okay, we have when we start the reaction, we have just the condition, the initial condition that they all are in end state. That is the enzyme state.